Good day everyone, my name is Patricia Apio and I'm going to present on molecular docking, an identification of glycogen synthase kinase 3-beta inhibitors for amyotrophic lateral sclerosis. So what is amyotrophic lateral sclerosis? Amyotrophic lateral sclerosis is a neurodegenerative disorder that affects motor neurons that are found in the brain and spinal cord, causing them to weaken and die, resulting in poor muscle management, characterized by muscle weakness, atrophy, and/or paralysis. The mechanisms of the neurodegeneration include oxidative distress, mitochondrial dysfunction, and neuroinflammation. The rationale for targeting glycogen synthase kinase 3-beta enzyme. So we see that this enzyme is upregulated in the neurons of patients suffering from amyotrophic lateral sclerosis. We have also observed that in many of these patients, there is a formation of a toxic complex agglomeration of TDP43 protein. And this agglomeration of the protein results from hyperphosphorylation, which causes them to stick together, causing a toxic complex that leads to motor neuron death. So the GSK3B enzyme is responsible for regulating this phosphorylation. And when we inhibit the enzyme, it means we stop the phosphorylation, and then we shall not form neurotoxic complexes in the neurons. Yes. So the study objective was to evaluate selected GSK3B inhibitors using molecular docking to identify potential candidates for repurposing in ALS and to compare binding affinities at the ATP binding site. The methodology: we obtained the protein GSK3B from UniProt and Protein Data Bank using the following IDs. The protein was prepared in BioVia by removing water and ligands and addition of polar hydrogens. We prepared the ligands in PyRx by minimizing the energy and converting them to PDBQT files. Thereafter, the protein and ligands were docked using AutoDock Vina in PyRx. The protein interactions were analyzed using PLIP online software and visualized in PyMo and BioVia for the 2D and 3D molecular interactions. The docking results: from our 5 candidates, we realized that elraglusib had the more negative binding energy score, making it the best candidate from molecular docking. It was compared with the second best candidate, which is SB216763, to see if they are binding to the same active site, as well as with tideglusib. Tideglusib is already a drug candidate studied for the management of Alzheimer's disease, where it is shown to show neuroprotective properties, and that means it has been studied for its ability to cross the blood-brain barrier. So comparing elraglusib and SB216763 showed that the top 2 bind to the same active site, and from the 2D and 3D interactions of elraglusib and our protein, we observe that there are hydrogen bonds, van der Waals bonds, and covalent bonds, showing a strong binding affinity with the protein active site. In the interaction analysis, we see that the hydrogen bonds are between leucine and histidine amino acid residues with the ligand, and the hydrophobic interactions are observed between isoleucine and proline residues. All ligands showed a favorable predicting binding energy. However, elraglusib demonstrated the strongest docking score. And since elraglusib is already a drug candidate in clinical trials for the management of cancers, hepatic cancers and blood cancers, it gives an advantage because it already has pharmacokinetic and pharmacodynamic data that can be used in the study to repurpose elraglusib as a potential candidate for the management of amyotrophic lateral sclerosis. Additionally, since these docking scores are computational, in vitro studies are needed to confirm the suitability of elraglusib in the management of ALS. Thank you.